Hi, Eliza, how are you?
I'm good. How are you?
Good.
I forgot for a second, I forgot it was Ash Wednesday. And so I saw the thing on your head and I was like, wait, what'd you put on your head? And I realized it was Ash Wednesday.
Like, girl, you got a little something up there. Yeah, I just got back from that mass, so they're fresh. It's kind of funny because afterwards you kind of like look at each other and you're like, so what kind of, did you get a smudge or did you actually get a cross? It's so funny to me. All right. So to the lucky few that got a couple or 24 hours extra to study for this exam, or not exam, the quiz, I'm going to be finishing up chapter 7. I think everybody here was able to make it to Mondays. I want to say, let me double check that. All but one, but that's okay. That recording is posted. That is 6 and 7 strictly quiz review. Today I'm going to be leaving off, picking up where I left off for chapter 7, and then I'm going to go over the dilutions. I know that was a little bit tricky because of the wording, but I'll be working on the dilutions with you guys, and then I think that should only take up about 45 minutes. We'll stop for questions in between, and afterwards, I'm gonna probably also talk to you guys about the operons, but I believe that's part of chapter 7. So I'll go ahead and start that. Any questions before I start? Good? Okay, cool. All right. Go ahead and start the presentation here. Why is the forehead so big? Okay, there we go. So I got a pretty decent way through here. I remember strongly emphasizing the semi-conservative, bidirectional, and semi-discontinuous, those 3 words. I'm not saying that there will be a question like that on the exam, but there very well could be a 3-point question like that on the exam, and it's looking for those 3 words and maybe the definitions. Okay. So we talked about transcriptions and the 3 stages, right? The 3 steps, the elongation, the initiation, elongation, and termination, right? I think we stopped right about here. Yeah, I said that this was redundant, so that if we did have any mutations, that that there was a less likely chance for it to actually express itself as harmful. Let's see. I talked about tRNA and how the tRNA is the anticodon and the codon is the mRNA. The anticodon is complementary to the codon, right? And the anticodon isn't what you look for in terms of the actual amino acid. So like if we were to be looking at this table, we wouldn't be looking at the tRNA anticodon, we would be looking at the codon itself. Does that make sense? Okay. Remember talking about that. And then I remember talking about the ribosomes and how they're fed through, right? The codon, the mRNA is fed through the ribosomes, and these amino acids with their complementary anticodon come in through here, right? And bind to the correct site, right? The A site is where it kind of reads it. The P site is where it actually binds. And they kind of just shimmy down. Let's see. Yep, they just shimmy down, form peptide bonds, right? And then this tRNA section snaps off and leaves this peptide bond between amino acids. This is probably a great video that I can't see, but we can see how it's not just one ribosome at a time, right? It could be several along the same strand of mRNA. And know that transcription and translation both occur in the cytosol. Okay. I'm gonna try to see if there's anything else. Okay. All right, this is a video. I think it's just a short one, but yes. Now this is where it gets to mutations, right? So genes are expressed all the time, most of them, and when they're transcribed and only translated when they're needed, this conserves energy, and it is an anabolic reaction, right? When we do that, quorum sensing is the regulates production of some proteins, right? And if we regulate the production of proteins, that means when do we turn them on and when do we turn them off? Let's see. So this gets to the idea of the operon, right? So we have our inducible operon and our repressible operon, I want to say. Yes. So we have inducible, which is an example of the lac operon, and then the tryptophan operon is a repressible operon, and I'll show you guys how those work in just a second. Okay, so this is the lac operon. Watch this video, and I would recommend drawing it out. So what happens when you have an inducible operon is this is the saving energy thing, right? The DNA has this gene, and it has a promoter and a controlled region, and then some other genes, right? That's kind of its structure. In the presence of specific proteins, so like lactose, in the presence of lactose, it will trigger this gene, and it will induce the gene to produce lactase. So in this video, you'll kind of see how it works and step by step what happens. This is also a good drawn out version, right? So we have the repressor mRNA that constantly represses this gene, and it is continuously translated so that this gene is not being expressed at all. All of a sudden, we have it's specifically allolactose from lactose, but lactose gets presented to our body, and a monomer of lactose comes in and attaches to this repressor, which initiates the continual translation of this repressor mRNA. It attaches to this repressor and binds and makes it inactive, right? So it no longer can make that continuous translation happen, and it can no longer translate that repressor mRNA, and it can no longer repress the gene, which means that this is going to now produce lactase, right? So the best analogy I can give for this is, you know those cars you had when you were a kid? If you pulled them back and held them and then let them go, and they go really far, right? So imagine you're in that pulled back state. Your hand is the repressor. You're preventing that gene from being expressed. All of a sudden, the presence of lactose binds to that and your hand is now bound to something else, and it lets that gene go. So that would be an example of an inducible operon. Okay, that's the lack operon. So now to move to repressible operons, right? It's just the opposite. So you can watch this video, but it's also summarized right here. So this is a gene that's constantly expressed, right? It's being constantly expressed, and there is a repressor, but in order to repress, it needs to be binded to something. So in its normal state, the mRNA is being translated and it's promoting the operon. It's letting the operon go constantly. It doesn't matter what's in your system, it's going to be going. As soon as tryptophan comes in, it binds to this inactive operator, making it active and binding to the operon, making the operon itself inactive and that gene no longer expressed. Does that make sense? It's basically just the opposite, right? They're just opposites of each other. Okay. Now, the reason why this is so important is because you guys just did the P-Glow lab. And the P-Glow lab, what do you think that's an example of? Which type of operon? I gotta check. Yes, so the proper name for the type of lack operon would be yes, there you go. Alrighty, so the P-Glow lab shows you an inducible operon, right? You guys will see that in lab on Thursday, or you guys already saw it today. And we will go over specifically what plates do what and how it functions, okay? I'm just gonna get through the rest of this first. You guys know the difference between genotype and phenotype. I'm sure you guys do. What we need to memorize is the mutations. So there's two types, point and insert or deletions. Those are the two bigger categories. Inserts and deletions are pretty simple. You either insert one on accident, it could be any of them, or you delete one. And think about that just for a second. You know, if you have all these base pairs coming in, all of a sudden your DNA is thrown completely off. There's no saving it because everything's going to be affected. Every 3 is no longer going to be what we thought it was. So those are harmful. Those are the most harmful. Point mutations are a little bit different. So we'll go through those right now. Let's see. So understand that point mutations are, let's look at this AUG on the mRNA right here for the methionine. Let's imagine that was changed to an AUC, right? AUC. That would be an example of a point mutation. It's taking one base and changing it somehow. The insert or deletion would be, instead of there being, let's see, 3, 6, 9, 12, 15, instead of there being 15 bases here, there'd be either 14 or 16. All right. So let's see. Okay. I'm gonna go here. So silent point mutations, what essentially happens in silent mutations is nothing. Nothing happens. It codes for the same thing. The redundancy of the chart is on purpose. Our body, if there was to be a mistake, wants us to cover our butts, right? If there were to be a situation where, I'm going to give an example. Let's say that we mistranslated, our DNA mistranslated, right? And instead of it being GUU, that's the lower left box, instead of it being GUU, all of a sudden we have GUC. It still codes for the same amino acid. That redundancy does nothing, right? Another example is on the side there. But that redundancy allows us to not have any detrimental effects from a mutation like that. Let me see. And the whole idea is that it's silent because it's not expressed in the phenotype. It's only in the genotype. Okay, so missense mutations. So this is a substitution, and then it changes the codon. So it actually, the redundancy, it's fallen through the cracks, right? So, oh no, this stuff's falling. Okay, so it's fallen through the cracks, and we've changed this amino acid, right? And that can mess up our whole protein. And sometimes that affects the function of our protein, right? Because structure equals function. So these are pretty bad too, if we were to change up a point mutation on a missense mutation, right? So missense is just a mix up, right? Nonsense mutations. This is when, you know, I'm going to try to think of a good example. Let's say you're starting a race, right? And all of a sudden somebody comes out and says, stop. It's like, no, I'm supposed to be like running here. I'm supposed to be going as fast as possible. In fact, you're telling me the opposite of what I'm supposed to be doing. This is what the amino acid sequence is feeling, right? There's a start codon. I might go for 1 or 2, and then all of a sudden it's telling me to stop. This isn't normal. This isn't supposed to happen. It's the premature termination of the protein, right? Of the amino acids. So nonsense, think of that race and all of a sudden somebody telling you to stop. Okay. Let me see. One important thing to know about this, almost all nonsense mutations end with a non-functional protein. I think she wants you to know that. Okay. So another word for so let's see, another word for deletion is frame shift, but I don't think it should be under point mutation. No, I guess it is. Okay, well then there is a little bit of oh, okay, I see. So frame shift point mutations are specifically the insertion or deletion of only one. So if she were to ask you on an exam, what type of mutation is the deletion of 2 base pairs? Your answer would be an insertion or deletion genetic mutation, not a frame shift. So frame shift is specifically the insertion or deletion of just 1. Otherwise, it's an insertion or deletion mutation. Those are the worst. Those are the worst, because look at all this. You have those amino acids on the bottom, and only one, I think, let me see. Yeah, only one stays the same. Right? Let's see this one. If you look at the normal one versus the insertion one, 1, 2 stay the same by chance, because that redundancy might save us, but you never know, especially not on a 300 base pair long amino acid sequence, right? Something like that, if there were an insert or insertion of deletion, especially of more than one code, like more than one, then all of a sudden we're all out of whack, right? Let's see. So that's all the mutations. Any questions on the mutations? I look like a ghost in the sliding. I'm sorry. Okay. So moving on, if there aren't any questions, I did want to briefly cover sickle cell as well. It's a type of mutation. You don't need to look at this and tell me where's the beta chain, where's the alpha chain. All you need to know is that it's a, it's just a conglomerate. I think it's called a tetra. I think it's tetra protein or something like that. And it carries hemoglobin, right? Or it carries oxygen in this hemoglobin molecule. So what you need to know about sickle cell is who it affects, not how many. You'll just need to know really who it affects. And let me see. A few of the symptoms. If you were to be given a question, what missense mutation results in sudden pain and frequent infections and affects the hemoglobin. You should probably think of sickle cell anemia, right? And it basically what happens is it just renders these just not functional. That's one of the biggest threats of mutations, is that they're just rendered non-functional. And if we don't have certain proteins, we can't do certain things. So any questions on sickle cell, anything at all? I'm gonna go on here. I'm trying to think what she was trying to do here. Oh, this is more of the specifically what is mutated. So in this case, glutamic acid should be at the 6th position, which just means it's 6th in line. But instead, we have, I believe, valine. Yes, maybe, or maybe there's just 2 switched. Well, it's a missense, so just know that it's glutamic acid, I suppose. I don't think I ever was asked specifically what it was, but if she were to ask you a question that was like, normally there should be this amino acid in this position. What is there instead? Just know how to find the position. And then of course how to read that chart, right? You will definitely have to read the chart on the exam in one form or another, whether it be lecture or lab or both. Let's see. Okay. Ooh, malaria. We love malaria. Oh, yes, this is very interesting. I just remembered. Okay, just by looking at this. So there's a very strange thing about malaria. Malaria actually reduces, or let's see, malaria isn't as infective if you have sickle cell anemia. So if you have sickle cell anemia and you got bit by a mosquito that had malaria, you have a higher chance to not get malaria than somebody who doesn't have sickle cell anemia. It is a weird genetic kind of upper hand that you somehow have. You're still kind of losing because you have sickle cell anemia, but you won't get malaria, or you're less likely to. It's very interesting. it inhibits the cycle that malaria has, and you'll learn about malaria later. It's very interesting. But yeah, that's the gist of that. Okay, so I think she's added these, or I don't remember these that well, but definitely know the difference between spontaneous and induced. I think you know the difference between at least the two words spontaneous and induced. So let's look at spontaneously induced mutations, right? It just kind of happens, an oopsie, if you will. Our ribosomes don't do something right. Our DNA is mistranslated. Something happens, right? It's out of our control, right? It just, oopsie. So with a spontaneous mutation, it's, you know, it's up in the air how it happened, why it happened, and what it did, right? But induced mutations, this is why you always wear your sunscreen. Radiation, even from the sun, can cause DNA mutations. And these are induced. The radiation induces mutations in your DNA. This is why skin cancer is caused by radiation. Radiation changes the way DNA is synthesized, and it is a lot easier for a cancerous cell to be produced somehow and then reproduce, right? Radiation would be an example of a mutagen. And let's see. I think that's just about it. That's all you really need to know about mutagens. Just know that also, radiation and chemicals. For those who did the oxidase test today, right? The TMDP, that is a mutagen, right? It can cause serious harm to your DNA. Okay. Hang with me. You guys know, I already talked about radiation. This is what it causes. Thiamine dimers. Be very acquainted with this word. Thiamine dimers is where instead of a bond between an alternating peptide, it binds between two thiamines. Okay. And we know that the GC content in prokaryotes, right? The GC content in prokaryotes is higher than us. So that means in the inverse that we have a higher AT content. So one of these dimers could very well be produced by radiation. Let's see. Yeah. So there are certain ways that we can just somehow proofread and fix this DNA. So let's see. What is the... So there's a thiamine dimer. So endonuclease breaks the DNA, right? It chops out the dimer. When it chops out the dimer, DNA polymerase comes back in and fixes it. And just like in DNA replication, ligase is going to come in and glue those pieces back together. And we're going to discard the thymer. We don't want it. The dimer. The thymer. The thiamine dimer. This is a great video to watch, and it shows you kind of the process a little bit better. Yep, skin cancer. Another one is the xeroderma pigmentosum. That's just another form of defective DNA. Let's see. Ooh, we got the sex pilus. Okay. This is sometimes a hard one to wrap your head around. I know it's hard for me. The difference between vertical and horizontal gene transfer. So horizontal, right? We are going at the same plane. Think of a graph as the X is kind of just, I don't know, forget I said X, just the Y, right? The Y is time. So we start at, 0 time and move to a greater amount the lower we go. So when we have horizontal gene transfer, right, we have one cell here, and at the same point in time will transfer DNA to this cell, and suddenly they will express the same gene. That is horizontal, right? Horizontal as in the horizon. When we have vertical gene transfer, we have a parent cell and 2 daughter cells that receive this DNA, this information. Usually this type of information is for a selective edge, right? It's survival of the fittest. Okay, so the difference is different generations between horizontal versus vertical, and the process of giving it, right? When a parent cell is giving 2 daughter cells DNA, it's through its own separation and fission, binary fission. It's through its own binary fission. However, the horizontal transfer of information is usually through a pilus. And remember, that was the smooth versus rough cocci experiment. This basically just says genetics cause diseases. Like the genetics of a bacteria cause diseases. Genetics controls everything about a bacteria. Of course, it's going to tell you how it interacts with other organisms, how it infects, how it might cause disease, symptoms, everything. What really gives it an edge is acquiring this resistance to antibiotics, most notable MRSA. There's also VRE, and you'll learn a couple more during your case study number 2s. It could show you different, like it looks at another organism and it's like, I'm gonna take that, right? So a good example is the E. coli 0157H7. This specific strain of E. coli has the Shiga toxin. The Shiga toxin is acquired from Shigella. Shigella is another one of those things that cause you dysentery. It loves to make you poop liquid, make you pee out your butt. So there is that. And then let's see. Oh yes, so if you are if you want to be an epidemiologist, it's very interesting how all of a sudden diseases can mutate to a point where they can infect different organisms, right? So if we have the bird flu can infect us. The bird flu has the potential of infecting other organisms, right? So that is one edge. Let's see. Sorry. Okay. So let me move on. And then yeah, this is basically it. We have our donor cell, our competent recipient cell, and this plasmid that has the genes of interest go to the recipient cell, and then bam, both have the same genes. This is through the process of transformation. You need to know all 3 of these by heart. They're pretty interesting. It's just kind of hard to wrap your brain around them. I'm gonna let you guys take a quick 5-minute break. Be back at 7:40, and I'll start diving into this, and then at a certain point I will transfer to the dilutions. Okay, you guys can go ahead, take a quick 5-minute break, and then I'll start up the Zoom again. Okay.